Thank you very much, Charla. The last speaker before we have the dinner break is Marianne Rasmussen. She has been the director of the University of Iceland Research Center in Húsavík since the establishment in 2007. She is going to tell us about the highlights of whale research at the University of Iceland Research Center in Húsavík since 2007. So, Marianne, please.
Sorry, it takes a while to open. Yeah, so finally, so I actually changed the title a little, so I will mainly talk about some of the recent research projects. And I picked some different examples of some from the last few years. So the first one is that probably many of you know that we have been tagging the whales for quite some summers now, and so I wanted to start with the example of the humpback whales and see if this one is working. Yeah, so this has been done together with Tom Akamatsu from Japan and a colleague from Denmark, Mikkel Willum. So Mikkel has he's bringing the pole called a Willum pole that we use for the tagging, and Tom Akamatsu is bringing the tags that we put on the whales. And then, as you see, we have this platform in the front where it's easy to stand and easy to put the tag on the animals. And one year we put camera tags on the humpback whales, and that was part of a Japanese Ph.D. student. She just finished, but I'll just show an example of that also. So this is the view from the humpback whale with the video camera on the back. And so besides the video camera, we also have the time depth recorder and acceleration. So this is a schematic drawing of what's happening when the whale is diving and doing these lunge feeding underwater. So all the red, that's when the humpback whale is opening the mouth and taking in. And in this case, we could see on the video camera that the humpback whales were mainly feeding on krill in June. So what she did, the Ph.D. student, she went through the video frame by frame, and like on the B picture, you see all the dots, that's actually krill. So she was making drawings around all the krill and then could estimate how much krill you could see, or how much krill the humpback whale was feeding on. But it was not only the prey you could see, you could also, in this case, you could see another humpback whale also in the view from the camera. And what was interesting was that the feeding duration, then it was shorter in front of other animals. So if other humpback whales turned up in the view of the camera, it seems like the humpback whale that was feeding was interrupted in the feeding, and the feeding duration was shorter, and it was less lunge dives. Then I jumped to another project, still on humpback whales, that we did also out here a few years ago, together with another PhD student, Bertrand Bouchard. He's from France, and he also just finished. And for that, it was to look at behavior responses of humpback whales to food-related chemical stimulus. And the picture A, that's the experimental setup. And the first one, that's where the chemicals were put in. And then we have the other one, that was the platform for recording both. There was a hydrophone to record the vocalizations, and then we had GoPro cameras, underwater cameras. And then from above, we used a drone also, so we were filming the behavior also using the drone. And so what we tested was we had krill extract, and then we had a control that was kind of orange clay that had the same color as krill, but obviously without the taste or the smell. And then we tested the DMS, dimethyl sulfide, that it has previously been shown for at least marine seabirds that they can detect that, and so we wanted to see if that was something the whales could detect also. And this is a table showing the different behavioral response we looked at, and also part of the data analysis. And finally, to the results. So this was tested also in Madagascar and in Antarctica, but I'll just point out the results from Skjaldvangi Bay. So there was a difference between the krill extract and our control. So the humpback whales were staying longer when there was the krill extract. So it seems like that they could detect it. So that was the conclusion for that. But on the other hand, the DMS and the control, there were no difference between these two. So the humpback whales could probably not detect the DMS. And then I go to another project that we will do now this summer in June again for the third time, and it's together with the group in Germany, University of Hanover, and we have been testing what's called a marine vibrator, and that's the loudspeaker you see to the right. It's 850 kilogram, so quite heavy. And with that we play low frequency sound in the same frequency range as the blue whales are communicating. And in order to do that, we needed a fishing vessel with a crane, so a big enough crane that could lift this loudspeaker. We had one from here the first year, but the boat almost sank, or at least it flipped over, so we could not use that boat. So this boat was sailing up here from Keflavik, and it's coming back in the summer for the same project. And before playing these the sound, before actually doing the playback, we put out a hydrophone array, and that's with 4 sound trap recorders that are with the buoy on the surface buoy on the top, and then we have them spaced either 500 meters or 1 kilometers apart. And we use that when the whales are vocalizing. Then we can afterwards, by using the difference of arrival time to each of these, we can calculate where the whale was when it was vocalizing. And then, of course, the tagging. I just wanted to show an example for the blue whales too. So this is Takashi, also from Japan. So this is our results so far for the 2 field seasons we had. I mean, so it's both been blue whales and humpback whales. And as you see, first we do like a pre-exposure, so that we get the baseline data on how the whales are diving and also how they are vocalizing. And then we start the exposure, so that's when we are playing back the low frequency sound with the loudspeaker. And we do that for about an hour, and then there's the post